Samantha Paramal, and my student independent investigation topic is on inherited Palmoplantar keratoderma. The question I was investigating was what ion channel and what mutations have been linked with this disease, and what is the evidence? What is inherited Palmoplantar keratoderma, or PPK? Palmoplantar keratoderma, or PPK, is characterized by a thickening of the skin on the palms of the hand and the soles of the feet of affected individuals. There are different classifications of hereditary PPK, and there are three main factors that allow you to classify PPK. Number 1, the specific form and structure and distribution of PPK. 2, the presence of associated skin and other tissue disease in sites other than palms and soles. And number 3, the presence or absence of fragile or blistering skin. I will now briefly go over the different types of inherited PPK. The first type of PPK is diffuse PPK. It becomes present in early childhood, initially starting with the redness of the palms and soles. This gradually becomes thicker and develops a yellowish, waxy appearance which can be seen through the images on the screen. The second type of PPK is a focal. Similar to diffuse PPK, the abnormalities begin to appear in childhood. Focal PPK is a rare condition compared to diffuse and punctate PPK, which I will go through in the next slide. Focal PPK is the development of painful blisters and calluses on the soles of the feet, mainly, as it's not very common on the palms of the hands. As you can see in the images, the blisters and calluses are quite large and are also yellow in appearance. The third and last type of PPK is punctate PPK. Unlike diffuse and focal PPK, punctate appears in the late childhood and early adulthood. It causes tiny bumps of thickened skin on both the palms of the hands and the soles of the feet. From the image, it just looks like large sections, but each section is made up of these tiny bumps of thickened skin. And similar to the other types of PPK, it's also yellow in appearance. So what ion channel is linked to inherited Palmoplantar keratoderma? The ion channel linked to PPK is the water channel protein or aquaporin 5 or AQP5. Aquaporin is a part of the family of cell membrane proteins. The main function of AQP5 is to allow the osmotic movement of water across the cell membrane. Aquaporin can be subdivided into two groups. The first is aquaporin, which is only permeable by water, and the second is aquaglyceroporin, which only allows passage to other small solutes, in particular glycerol. AQP5 comes from a subgroup of aquaporin and has a high water permeability. Aquaporin 5 is distributed in the apical plasma membrane and is known as an exocrine type water channel protein because it is involved in the excretion of water from exocrine glands. I will now go to the mutations linked to inherited Palmoplantar keratoderma. I will mainly be focusing on the mutations linked to aquaporin 5, but I just want to mention that they have found other mutations linked to inherited Palmoplantar keratoderma. So mutations in KRT6C gene and the KRT16 gene have been found in affected individuals with PPK. The mutations prevent proteins to interact with other keratins to form strong, stable networks within cells. This type of mutation has particularly been found in focal PPK. Other mutations have been found in AAGAB, which encodes the alpha-gamma adaptin binding protein P34, which plays a role in membrane traffic as a chaperone. This particular mutation has been found mostly in punctate PPK. So as I said, I'll be focusing on mutations AQP5. Missense mutations have been found in aquaporin 5 in families affected by diffuse non-epidermolytic PPK or NEPPK. The characteristic associated with NEPPK is that it involves increased water uptake. The mutations are thought to cause the water channel to stay open in a cell membrane and conduct water continuously. Through Sanger sequencing, they have been able to track 5 missense mutations identified in aquaporin 5 in the families affected with PPK. So below is the 5 graphs from the results, and I will briefly go through the amino acid change that occurred at each graph. So in the first graph, it showed that isoleucine at position 177 was changed to amino acid phenylalanine. The second graph shows that isoleucine at position 45 was changed to styrene. The third graph shown on the screen shows that alanine at position 38 was changed to glutamic acid. The fourth graph shows that at position 188, that the amino acid arginine changed to cytosine. And finally, this graph shows that at position 123, asparagine changed to aspartic acid. I will now go through where these specific mutations occur on the aquaporin 5 and what type of effect they have on the water channel protein. The first three mutations I will speak about is isoleucine at position 177, which changed to phenylalanine, isoleucine at position 45, which changed to styrene, and arginine, which changed to cytosine at position 188. These mutations are located at the residual lining of the extracellular end of the water channel, which can be seen through the image on the screen. They are all thought to have direct impact on the water conductance of the aquaporin 5. So when the isoleucine changes to phenylalanine, it changes the diameter of the protein, as phenylalanine is a much bulkier amino acid than isoleucine through steric hindrance. When isoleucine changed to styrene, the effect it had on the water channel is that styrene is a smaller amino acid than isoleucine, which therefore causes the diameter of the constriction point to widen. And the last mutation, when arginine changes to cytosine, the effect the substitution has on the water channel is that the smaller cytosine causes a widening of the channel at its narrowest point. Also, arginine at position 188 is a key component of the constriction point in the water channel and is the most highly conserved of all the AQP5 amino acid variants identified. The other two mutations, alanine, which changed to glutamic acid, and asparagine, which changed to aspartic acid, are located in the extracellular surface of the water channel protein and can be seen on the image on the screen. These two mutations are also thought to have a direct impact on the water conductance, even though they are both located outside the water channel itself. Both of these variants are situated in the extracellular loops, which are important for the stabilization of the tertiary structure aquaporin 5. The peptide backbone of alanine at position 38 and the intramolecular interaction of asparagine at position 123 both form bonds, specifically hydrogen bonds, with residues on helix 6. Due to the amino acid changing to a charged side chains, glutamic acid and aspartic acid is thought to most likely cause the position of helix 5 to change in the proteins with the mutation. Thank you for listening. Here are my references. Thank you.